uh, towards the end of the day, it's going to be very difficult for digesting scientific facts, but I cannot help talk science, unfortunately or fortunately. So let me get straight to the point. See, I started off the work. I'll just start a quick intro about the story. I mean, the way I started researching Ayurveda. It's been 32 years since I've been working on this, and as a medical student, I figured the pros and cons of the modern medicine. And then while modern medicine, I immensely respect modern medicine, by the way. I love Ayurveda. So I'll tell you why it is so, and you will quickly see the data that has generated over the last 32 years in treating patients in arriving at mechanistic correlations of Vata, Pitta, Kapha. That's going to be really interesting. Why? Because, we talk about genomics. Friends, there's something called genomics and epigenetics. Everyone would understand, right? Now, genomics, from the data that we have, 92% of times when a gene is expressed, you don't find a corresponding protein in the body. 92% of times. But the converse is true. If you find a protein in the body, you'll always find the gene expressed. So why is it so? That's when, Singh et al. In 2003, when he came up with the SNPs, he explained, and several others, joined that just before the Human Genome Project, or just after the Human Genome Project was unraveled, they figured that not always when the DNA, when the DNA or a gene translates, it doesn't always transcribe into a protein. So why is it so? No one knows. So that's why there's something called translationomics. In the subfield of genomics, you have translationomics, you have transcriptomics, and then you have proteomics and metabolomics. Just because you find a protein in the body or in the blood, it doesn't always act as it doesn't perform its function. And hence, metabolomics, that metabolomics is a science of how the metabolic pathways get affected in the body under the influence of these proteins, interleukins, and so on and so forth. So for me, as a student of biochemistry, I love biochemistry. I never forgot any of the basics of chemistry or organic or inorganic, and I love to equate the Vata, Pitta, Kapha to the buffer systems in the body. These are the Vata buffers, Pitta buffers, and Kapha buffers. That part came to me when I was reading the story of a Russian cardiologist, actually Mechnikov, in the earlier century, in late 1890s. He suggested that there could be different buffer solutions in the body, and why a buffer is called as a buffer, because they don't change their pH so easily. So this Russian scientist, who had been my professors, my professor who taught me immunology, his boss, his guide, he suggested that the cardioplegia could be achieved, and a new cardioplegic solution could be achieved by exacerbating the kapha properties. That's what he said. And that's documented. That was way back in 1930s, friends. Kapha induces sluggishness in the body. When you're able to induce sluggishness in the functioning of the heart stops. And that's exactly is called as cardioplegia. Used as a cardiac surgeon, that's what we do before we do an open heart surgery, or we do an intra-cardiac repair, a TOF or a ASD or a VSD. We have to stop the heart, then do all the repair, and then restart it. So what I'm trying to say is, several scientists across the world have been researching these concepts of Vata, Pitta, Kapha. So having said that, and I felt the necessity for correlating the doshas and the subdoshas to metabolomics. So that's my journey through metabolomics, and how I figured that, I deduced that, it's an end stage disease that has to be targeted, because that's the area where we could work together. Ayurvedic doctors, allopathic doctors could work together, because we commonly have the problem. Ayurvedic doctors, as well, I'm a BMS doctor as well. I just gave my second year exams in Varanasi. I took the NEET, and in 2017 they didn't allow me. I went to the Supreme Court to get an age relaxation to write the NEET exam, and I got admitted in BAMS. And so, because wow. Yeah, and I'm a Ayurvedic doctor, a naturopath, a data scientist, a Python programmer. So I think, if someone could volunteer, I would show the power of the metabolomics And I'll show how a rough estimate of your blood biochemistry could be done with the device I came up with. Any volunteers, please? While he figures out the volunteers, I'll just quickly go to the topic. Please, next slide, please. So this journey, as I mentioned, is all about correlating the concepts of Vata, Pitta, Kapha, or the Ayurvedic concepts to the modern science. See, the problem is, if there are two speakers, one speaks only in English and other one only in Russian, you end up with a Ukraine war. So he doesn't understand English, this guy doesn't understand Russian. But I keep, whenever I go to the US, they stop me. They said, you went to Russia. I said, so what? No, you're coming to US. I'm a doctor. I'm a global citizen. I'm here to heal everyone. So I always have this problem with immigration. That's because, I keep telling them, see, you guys don't know Russians' mindset. If you allow me to be your negotiator, I'll stop the war in an hour or so. That's what I keep telling the immigration authorities. So the thing here is that while some of us as Ayurvedic doctors get motivated to say that this is a different science, a complete epistemology, I slightly beg to differ. I would say that it's not a difference, but I see these two sciences as two sides of the same coin. So let me unravel the bridge that that I try to actually connect to these two sciences and then come up with a lot of data. So I'll next slide, please. So we applied all allopathic research methodologies to unravel the science of the dosha expression. Keep going. Next slide. Next slide. So and through correlation of these, creation of models of Vata, Pitta, Kapha in experimental animals and their subdoshas, I tried to correlate to the mechanical or the mechanisms, or fundamental mechanisms that essentially that are guided by these doshas, and we generated a lot of data, more than 3,000 markers for Vata, Pitta, and Kapha quantification were arrived at, and then we applied that in improving clinical outcomes, and especially in the end-stage cancer patient, end-stage patients, please. Yeah, you can start. Anyone who wants to get his blood biochemistry non-invasively? No. Yeah, please. So this is not something new, friends, and I'm not inventing something new. Polyscientific Ayurveda. It's just that you go to an Ayurvedic Vaidya essentially. Because he's both a Sanskrit scholar and an Ayurvedic doctor, and I'm taking a cue from Sushruta Samhita, chapter 4, Sutra Sthanam, 8th shloka. Maharshi clearly mentions Eka Shastra Madhyano na Vidyat Shastra Nishtayam Tasmat Bahushrutam Shastram Vijaniya Chikitsaka. What it means is, just by knowing one Shastra, you'll not become a good doctor. That's why in future, several sciences will keep coming in. When they come in, the duty of an Ayurvedic doctor, Tasmat Bahushrutam Shastram, he says, Tasmat, and hence, you've got to be well versed with several sciences, and with the new sciences that will that are going to come, have to be correlated, and that's only when you'll become a good physician. Is that what chapter 4, 8th shloka, Sushruta Samhita, Sutra Sthana clearly mentioned? So that's exactly what I was trying. So what I figured was an Ayurvedic doctor, because he was also exceptionally well versed with those days of translational Ayurvedic understanding, which could be called as an allopathic doctor of those times, was able to properly diagnose the VPK42 fingerprint of an individual. And once you arrive at that, applying a lot of data. Look at, Ashtanga Hridayam. Maharshi Vagbhata clearly mentions that Panchavimshati Pratishateshu Vata Pradhanaka. So what he says is, there are approximately 25, as per the population demographics of those times, he documented public health policies, that 25% belong to Vata Pradhana category, 25 to Pitta, and 25 to Kapha Pradhana. And about the mixed Vata, Pitta, Pitta Kapha, and Kapha, Vata Kapha were only 3 to 7%, 5 to 7% of people belong to that category. And only 3 to 5% of them had Sannipataja, which means they had Vata, all the three doshas played up like many of the scientists, they go crazy. So, but then, is that true today? Can we apply that today? I don't think so. Our data doesn't suggest so, because after understanding the markers of quantification of Vata Pitta Kapha, we figured that today, at least in my own hospital setup, in the last 22 years, after treating about more than 28,000 patients, and after doing a Nadi Pariksha of more than 400,000 patients, I figured that 75% of them have a BPK dominance in today's, and it is not homogeneous. It is not homogeneous inside his own body in different organs. If, say, for example, how am I able to say that? Because we have specific correlations of Vata Pitta Kapha to interleukin chemistry, to electrophysiological studies, to cell signaling pathways. That's what we unraveled, and that's what I want to say. What I'm trying to say is, when Maharshi Bhagavata explained the population demographics of those days, they have incorporated data science, applied math, and lab technicians. So what are the Mala Pariksha, Mutra Pariksha? A doctor, Ayurvedic Vaidya, was essentially a lab technician. And because he was also well versed with manufacturing practices, he could make, according to the Vata Pitta, if you encounter a Vata Hridroga, Vata mediated ischemic heart disease, you got to essentially take Arjuna terminalia, make a water extraction of it, and give the patient according to the Vata proportion in his body, or in the heart, if he's able to specifically understand the Vata proportions of Vata or quantification of Vata in the heart. Then if Vata led, or aggravation of Vata led to, or Samana Vata or Apana Vata led to the heart disease, then you use a water extraction of Arjuna. If it is a Pitta dominant or a Pitta Hridroga, then you use a Ghrita or a Asava. Or if you have a Kaphaja Hridroga, then essentially use a Arishta. And there are 16 different types of formulation: Vati, Guti, Kashaya, Arka. There are several formulations, 16 such formulations already mentioned. So because the Vaidyas were already well versed with phytochemistry of those days, of that language, they were able to formulate the medicine, and that is nothing but bioinformatics programming. So that is why I call this as a polyscientific Ayurveda, because all the current sciences, there are different sciences that are actually essentially involved in making an Ayurvedic doctor an Ayurvedic doctor. Next slide, please. So what we did was essentially, the crux of it, we know we don't have to go deep into next slide. Yeah, current problems in modern medicine. It was said enough, but I'll stumble upon I'll stop at this important slide to look at these different standards defining the guidelines for diabetes. If we say India is a diabetic capital, the problem is, according to the American Endocrinologists Association, the HbA1c should be below 5.5. According to the American Endocrinologists, it should be below 6.5. The American College of Physicians is coming up with a standard. It says it should be between 7 and 8, and never, see, this is important. They're at loggerheads with the endocrinologists and the diabetologists. They're saying it should not be below 6.5. Why? In their cohort, as per the trial done, ACCORD trial, they found that especially when a diabetic patient has concomitant IHD, 1 in 3 patients died when they were subjected to intense glucose lowering, pushing the HbA1c below 6.5. All right, so and then American Geriatric Association has its own standards. American Veterans Affairs Diabetic Association is saying, your HbA1c should be anywhere between 8.5 and 9.5. So that's the range of HbA1c that different standard bodies are actually processing. And luckily with the AI, a lot of metadata, it was analyzed, and then you got a lot of data, and then different standards. And by the way, these standards are all data driven. It's not just that they just wanted to give the standards to us and they came up with these numbers. No, they were all data driven. In all, I just pulled up all the 172 studies done by the Americans. And you know how many patients were investigated? 12.7 million patients were investigated, and about 5 million were retroanalyzed. And so the problem is, according to who are you diabetic is the biggest question. If India is a diabetic capital of the world, according to who are you diabetic? Because the veterans naturally are feeling hypoglycemic if you push their blood glucose below 7. That's the standard. Look up for the VADT trial. Veterans of his diabetic trial clearly demonstrates that these veterans had suffered hypoglycemia if the blood glucose was pushed below 7. And you already have the data from the Accord study. So there's so many problems that the current medicine definitely has. And then the problem with the statins is another one. Next slide, please. Another one that is really intrigued, because today we have data that suggests that apart from the Hunt study, apart from the statins controversy, okay, by the way, not that statins haven't helped. Had statins have helped about 54% of patients. 54% of people who have been put on statins have been saved. But the rest of the 46%, if you analyze them, 22% of them ended up with a new form of heart failure. You give statins to protect the heart, but that really failed the heart. New form of heart failure, liver failure, kidneys, interstitial tubular necrosis leading to chronic renal failure. Where the study published, it's a 92,000-page report done by Hunt and his colleagues from Norway. Not only that, today we have data that high amounts of HDL, see, high amounts of good cholesterol in Caucasians, Northwestern Indians, Baluch, Sindh, Pakistani, in this region around the Caspian Sea, they ended up with heart attack with high HDLs. And low LDLs also have caused heart attacks. Malinsky et al., they retro-investigated about 125,000 patients who were put on, who managed their triglycerides and LDLs below 100. And what they found was that 75% of the 125,000 patients who were retroanalyzed, who maintained healthy levels according to the American Heart Association standards, healthy levels of low LDLs, suffered from heart attack. So how do you manage? How do you target? How do you treat the cholesterol? It's the biggest, another big problem. There are other problems too, but it's not just about the problems. Of course, it's exceptionally good in acute care, emergency care. There's no doubt about it. But then there's fundamentally the data that they are coming up with is documented, and you can always pull up the data to understand the cohort and understand their doshic proportions. And then next slide, please. Keep going, next slide. So to understand this, I know this famous shloka, samadosha, samagnisha, samadhatu malakriya, prasannatma indriya manaha swasthya vidhyate. What it means is, we all know that when you bring about a balance of these three doshas in all of the 72 and 42 organs, naturally the agnis get normalized for that particular individual. The excretory waste always automatically is eliminated. The person is restored with physical, mental, emotional, social, and spiritual well-being, and swasthya, not just arogya. Arogya, again, based on Sanskrit syntax, roga abhasaha itihi arogyaha. Mere absence of disease is arogya, but swasthya is swasthanam shiryate itihi swasthya, which means that which confers you to the swasthan, gets back to you the original sthanam of swa. Swa in Sanskrit means atma. That which restores you, corrects you back to God, back to your own jivatma. The jivatma with the paramatma is swasthya. That's the beauty of this science, and then automatically regeneration of the tissues happen. We keep going, please. So what we did was, we pretty much keep going. Because this will be too much, So I just made a quick slide on the biochemical aspects of the Vata Pitta Kapha. So I'll not go deep into that, but because that's another synopsis from the entire Agastya Samhita. Next slide, please. Keep going. And what we did was that the key here is to identify the imbalances of Vata Pitta Kapha, quantify them, and appropriately give diets, give those diets that balance. That's why Bhagavan Krishna clearly mentions Yukta Ahara Viharasya Yukta Cheshtasya Karmashu. He never mentioned Su Aharasya, because Annam itself is Parabrahman, Annam is divine. So there cannot be good and bad food, there cannot be junk food. He said Yukta Ahara means that food which is appropriate to you, and that food which is appropriate to you is the one that balances the Vata Pitta Kapha in all of the 7 tissues and 42 organs. So that's why what we did was, that once you identify this, then you give the right food, the right exercise, the right pranayama. Say, for example, if someone has pitta aggravation, stop doing Bhastrikaras. If someone has a sympathetic aggravation, then stop doing Anulom, just do the Vilom. So there is a specific syntax of which asana, which pranayama to be chosen to each individual, depending on his VPK42 fingerprint. So the base of all of this is because the common denomination is Vata Pitta Kapha and their subtypes, the 5 types of Vatas, Pittas, and Kaphas. So what we wanted to do early back in early 2000s was, we wanted to create models of Vata, Pitta, Kapha. I know because I'm trained in the modern research methods, I could not be done away with those methods. So I had to apply those methods to understand Vata, Pitta, Kapha. So we created models of Vata, Pitta, Kapha by feeding animals, visitors trained rats, 15 in each group, with Vata aggravated, Pitta aggravated, and Kapha aggravated foods for Dvipakshe, as mentioned by the Charaka Samhita Sutra Sthanam. Dvipakshe is 2 fortnights. So what we found, next slide, please. What we found was, next slide. What we did was we measured, we isolated the hearts. Next slide. I will just come. Yeah, the previous slide. Sorry. Yeah. So what we measured after feeding these rats for the with these dosha aggravated foods for 4 weeks, we isolated the hearts based on a method called Langendorf method. And we put the hearts in a physiological solution. I mean, there's a Langendorf method. I don't have to explain that, but that's an elaborate process. I mean, I'll not be able to explain it right now, but you isolate the heart based on this method. The heart keeps functioning so that you essentially arrive at a reductionist method to check the action potential. So my target was if Vata, see, until then there was no proof of Vata, Pitta, Kapha existing, though, I mean, in the modern field. So we had to generate some effect of this Vata, Pitta, Kapha. So see, when I'm talking to you or when we talk to each other, what is happening? The sound is incident on the tympanic membrane. Tympanic membrane vibrates. On the inner side of the tympanic membrane, you have the auditory nerve, vestibulocochlear, nerve. And then that essentially generates, converts this mechanical tympanic vibrations into an electrochemical message. At the side of the tissue, I mean, at the side of the touch, the Pacini receptors convert the touch into an electrochemical message. So essentially, electric currents are generated. Action potentials are generated in the body at every level of cellular, biochemical, or your sensory level of interaction. So what was supposed was if Vata, Pitta, Kapha existed, they would essentially alter the electrochemical messages. I mean, the way the electric currents are generated. That was the hypothesis. So based on this hypothesis, we created the experiment. We did this experiment, and what we found was really brilliant. Okay, the heart muscle. What we did was we put in something called as a voltage active dye into the heart, and then that emitted electric currents based on the resting membrane, based on the change in the membrane potentials, and they were captured with computers, video cameras, and then we were able to capture the entire action potentials emanating from the Vata enhanced diet fed rats, Pitta, Kapha, respective rats. And what we found was the threshold membrane across all of the rats was -65. While the resting membrane was -84 for control. The control group of rats were fed with balanced diet as per the Russian Institute of Nutrition guidelines. So we found that the resting membrane was potential was around -84 for the control group of rats. We sacrificed one group of rats before the experiment and one group of the rats after they were fed for 1 whole month with balanced diet, and then there was pretty much not much variability there. But what we found was the Vata enhanced diet fed rats ended up with their resting membrane potential coming closer to -75, which was very close to the resting membrane. Pitta was around -90. Kapha resting membrane potential was around -108. What it means is really interesting because Vata, I mean, look at the difference. Why is this important is the difference between the resting membrane and the threshold membrane potentials is exactly the stimulus that should be given for that tissue to elicit an action potential, which means if you give anything less, it won't elicit an action potential. So the Vata diet-fed rats, Vata aggravated diet-fed rats, had a difference of 7. The Pitta had 25, and the Kapha diet-fed rats were 43. So the difference of these two. So what? It means that if you give an electrical stimulus of 53 millivolts across the entire group of rats, the Vata systems would elicit 7 action potentials, Pitta 2, and Kapha only 1. If you give only 1, I mean, if you give only 7 millivolts equivalent electrical stimulus, the Vata-fed rats would elicit only 1 action potential, Pitta and Kapha wouldn't actually produce any. So in comparison, go to the next slide.
This is interesting. You did this where in Russia?
In Russia, yes.
When was that?
It was in the year 2002.
2002.
Yeah, I came to India to just explain this in 2006 to CSIR. Unfortunately, I couldn't meet any of you.
Oh.
Yeah, we would have done a lot of experiments. It's really interesting. It was the first time.
Where is this, where is this, paper? Where can I get reprint of this?
I'll send it to you. It's there, online. Yeah. So this is for the first time, by God's grace, we were able to demonstrate the chalatva property of vata. What chalatva means is hyperexcitability.
So you are ahead of me actually. I thought
No, no, no,
you
no.
actually did this for the first time genomics, but of course you did these studies correlating.
No,
with
because
scientific-
I understood that this genomic study, as I mentioned, would not completely translate
So
into
you are right,
proteomics.
this is very-
That's why we went into straight electrophysiology, cell signaling, and metabolomics. So chalatva is for the first time uh, demonstrated in an experiment. Next, the one of the properties of kapha is what? Kapha makes the tissue sluggish because it is sthira and sandra. Sthira, where did the property sthira come from for the kapha tissues is because next slide please. The difference between resting and threshold membrane potentials in kapha is -1 or 165. So it requires larger. So I used to say earlier, so I respect all the politicians, but I used to say these are Manmohanized cells. So they just sleep over under the entire demon. I mean, until okay, I really respect this person. I just give these analogies so that we kind of remember. It's just an analogy.
Never.
No, not that. I'm not. Not because of that.
Sure.
I know, God. So these are like I used to say the vata cells are like Kejriwala-ized cells. Anything he goes on a dharna. He forgets that he himself is the chief minister. He can do several things on his own, but he doesn't. He just goes on a dharna. That's like Kejriwala-ized cells. And anyway, that's all. So the pitta cells are very interesting. So then we did several studies on subjecting these hearts with adrenaline and acetylcholine, the sympathetic and parasympathetic mediators. What we found was, interestingly, the adrenaline and acetylcholine on the vata and kapha rats demonstrated their effects. They increased the heart rate and decreased the heart rate respectively in vata and kapha, depending on their consistency at that time. But then pitta cells did not respond, or the response was insignificant, statistically insignificant. This is very interesting. Then I said, the first question that stuck to me, how are they eliciting action potentials? Because we thought action potentials would be elicited only when neurotransmitters, are subjecting their, these receptors are subjected to the neurotransmitters. But then, we dug out a lot of things and we found that apart from neurotransmitters, alpha-ketogluteric acid actually also causes elicitation of these action potential. And we measured the not mitochondrial alpha-ketogluteric acid, but intracellular. And you all know, if or modern doctors would know, alpha-ketogluteric acid is one of the metabolites of Krebs cycle. Krebs cycle happens inside the mitochondria, and that's a process in which the glucose breaks down. I have medical doctors here. That's very good. So the glucose breaks down in 3 steps. One is glycolysis, then the Krebs cycle, then the electron transport system. So what has happened is the alpha-ketogluteric acid concentration was 5 times in the cytosol, not in the mitochondria, in the cytosol, which means possibly, okay, this is not conclusive. We cannot conclude it. We have to do several experiments, but something that definitely suggests is that when pitta is aggravated, the mitochondrial activities is increased so much that alpha-ketogluteric acid actually escapes into the cytosol and elicits an action potential, because that is the one that produces action potentials as well. So that's why pitta people, they will not listen to you. They will listen only to food.
In one of our scientific reports, we have PGM1 gene is
Oh.
the signature for pitta.
Oh, right.
We
So
have reported that.
okay, right. PGM. Okay, yeah.
So PGM1 gene means energy. That's the energy gene.
Right, energy genes. Yeah, sure. So this is very interesting. So these are the people who will say, "Ghar mein ghus ke marenge." Matlab, I'm just joking. So the idea is that what we were able to deduce was the vata, pitta, and kapha were actually these doshas that are altering the metabolic pathways in which glucose participates. And by the way, all the diabetologists, if at all you're here, what I can say for sure is that glucose participates in more than 175 different metabolic pathways. And only 10% of these metabolic pathways are dependent on insulin. Adiponectin initiates about 12% of the glucose metabolic pathways. Incretin, I mean, see, we are forgetting that. I mean, when we go do these studies, we are forgetting that apart from insulin, there's several other, neuromediators. There are also humoral mediators, interleukin 10, interleukin 12, that also participate in your glucose metabolism and the regulation of the glucose. And then, so that's a huge study. Okay, by the way, this is a quick synopsis or quick teaser of my 545 hours of lecture of translational research. That's what I bundled up in the form of, bringing this gap. I wanted to create, help people bridge the gap. I demonstrated this to Shripad Naikji in 2014. But then unfortunately, they came up with a bridge course in which, they said Ayurvedic doctors can practice allopathy. While they were practicing anyway, they made it a political affair later, So the interesting thing, earlier slide, but I just want to stop. Take 2 minutes and then quickly show case studies and then end this because it's too much to. So what is happening is in Vata, okay, so what is at the end of an action potential, what happens is how does an action potential happen? Sodium ions, chloride ions get into the cell, potassium ions go out of the cell. So at the end of the action potential, sodium and potassium have to be exchanged to restore the ionic homeostasis, and that is an energy dependent process, ATP dependent. So which means if Vata cells are producing 7 action potentials, it's a hypothesis again, they are using up ATP faster, 5 times faster. Pitta, because they're producing ATP. See, the Pitta cells are actually producing 38 ATP molecules. So they're using only less energy because they're producing only 2 action potentials. Kapha, Manmohanized cells, they don't use up much energy. They don't need much glucose. So, and hence, Pitta, because they're burning up glucose faster, we found 5 times more alpha ketoglutarate acid inside the cytosol. We thought that their glucose demand should be 5 times higher because they are essentially, they're burning up everything, right? So the mitochondrial activity is 5 times faster, and hence the glucose demand is higher here. So the glucose demand for Vata is less because Vata essentially is triggering an electrophysiological exacerbation and not a mitochondrial exacerbation. So the glucose demand is only twice as much as the Kapha. So that's really why your ATP, your glucose need is different in different individuals. Without the context of Vata, Pitta, Kapha quantification, your efforts to reduce glucose, number 1 will be in vain, number 2 will be dangerous, as was demonstrated in the Accord study. I told you in the beginning of the story that, in this talk, that Accord study demonstrated that 1 in 3 patients, they died. When these patients, especially they had concomitant heart disease and diabetes, when they were subjected to intense glucose lowering, they died in the trial. The Americans had to abort the trial. American College of Physicians had to abort the trial. So after 2.5 years, when they got this retro data from their own patients. So next slide, that's fine. So one more concept. So what happens is when the glucose enters into a Vata environment, it gets acylated and ethanol forms. Either ethanol or lactate or pyruvic acid forms, and hence only 2 ATP molecules are generated instead of 38. But the same glucose, if it enters into a Pitta environment, it produces 38 ATP and a lot of carbon dioxide and H2O. And when the intracellular carbon dioxide and H2O increases, the partial pressures of these 2 gases, one gas and another liquid increases, they trigger the activation of carbonic anhydrase, and carbonic acid is formed from carbon dioxide and H2O. And carbonic acid, because it is an acid, Pitta is characterized by one of the properties called amla. Interestingly, this will be correlated again from these experiments, and Kapha, Sthira, and Sandra, because and hence your diabetic guidelines should be different for Vata, Pitta, and Kapha, and hence Kapha people, or if you find a Kapha dominance, then you need to push the glucose levels below 5.5. If you find a Vata person, you need to have your glucose levels between 7 and 7.5. You have a Pitta person, because he's burning glucose faster, his blood glucose could be, HbA1c could be anywhere between 8.5 and 9.5. I have several patients, I have treated more than 5.
Interesting, because our study which we have published,
Right.
we have shown that the DME loci,
Oh, these are okay.
drug metabolism enzymes, the fast metabolizers are predominant in
Pitta,
pitta.
of course, there should be.
There
There would
is
be.
a clear correlation. So
Absolutely.
what you're telling, but this study, where have you done?
Have you published it?
No, we haven't published
Oh.
it. We are just publishing it. We're patenting it wherever it is patentable. We're publishing it very soon, yeah. So this is unpublished, oh, the copyright is not there. So that is why your Vata diets and Pitta diets and Kapha diets will be different and should be different. And if you give statins, say in a Vata aggravated individual, so why is a Vata person producing more cholesterol? And this has been our observation as well. Vata dominant. I mean, there's no Vata predominant person today. In today's world, there are only VPK dominant people, 75% of them. Rest of the 25, they could be different, but when different VPK proportions and clear Vata dominance. I think you would find in your genomic studies as well, because you collected data of 6,900 patients, and clear-cut Vata, Pitta, and Kapha correlations were found only in 2,000 plus patients. So that's about only 1/3 of them had clear Vata dominance or Pitta or Kapha dominance. The rest of them had VPK. That's why you had to discard that number of patients' data. So that's another problem that we have from the genomic study. I mean, I'm not citing a problem, but that's how science progresses. We need to find faults in each of us so that we become better. So what is happening is like when you subject a person, I mean, when there's Vata aggravation, this person, if you don't give him enough glucose periodically, every 2.5 hours, this person is going to essentially this person, number one, could have different ramifications. Either there could be vasoconstriction leading to hypoxia and malnutrition leading to emaciation of the individual. Yeah, exactly. Yeah. So if in a Pitta, I mean, this is just one of the ramifications of this study. Pitta diets, for example, they have to be high glycemic index foods because they're burning glucose faster. So I tell you, patients come with, okay, I take up only end-stage patients or refractory diabetic patients. And all my 5,000 patients are refractory diabetic patients. Their HbA1cs are about 10, irrespective of they were put on 100 units of insulin, plus metformin, plus glibenclamides, I mean, all the sulfonylureas, biguanides, SGLT2 blockers, including, you name, all the drugs are, the gliptins, everything is in, I mean, after treating with all of this multi-drug, or polypharmacy, they still maintain HbA1c of less or more than 10. That's when they come to me. And Pitta individuals, I tell you, what I did was, first time it was very difficult because, it's very difficult to convince. I started off this work 22 years ago, and in a country like Russia, that's where I studied and that's where I practiced. You know what happens if you don't comply with any of your promises in Russia? You will end up with a war. So I used to promise, I had so much faith in our rishis. I used to say, if I don't treat your heart disease, you can take my heart. I used to write agreements with them because at that time there was no data, and I had to tell this to my patients. And luckily, I'm not only with my heart, but I stole several hearts there. So next slide, please. So in a Pitta aggravated individual, the body is burning up more glucose. Your intracellular ATP is so high. When the intracellular ATP is very high, it automatically triggers the production of both incretin and glucagon. Which means if you don't supply the body with enough enough glucose, these patients will essentially trigger the gluconeogenetic pathway through liver metabolism. And if you say, for example, given the SGLT2 blockers, you're sure to induce a interstitial tubular necrosis of the kidney and AKI leading to a CRF. Several patients, we found that happening. And when you give them enough glucose, I used to sometimes, based on their VPK proportions, Pitta proportions, I used to even suggest 250 grams of glucose, straight glucose. And some people used to get 8,000 to 10,000 calories per day. And you know what, from 10.7, several patients of mine, from 10.7, it comes down to 7.5 after 5 months, 6 months when you do the HbA1c, because, it's only when you satisfy Pitta that Pitta Shamaka happens. Once the Pitta Shamaka happens, the body's need to burn glucose reduces, and automatically, normal glucose metabolism sets in, and then, these alternative pathways get,
Bluetooth
connect. I will in 5 minutes. What time? Yeah, no. In 2 minutes, yeah, I'll finish that. We'll do again, people will come tomorrow, maybe, right? We'll do this tomorrow. Yeah, Bluetooth is not getting connected. I'll finish this slide and then we'll go. Yeah. So next slide, please. Next slide. Keep going. So everything should be personalized. So what we say, I just mentioned, more than 3,000 markers were quantified for Vata Pitta Kapha. Not only that, possibly we were the only ones to credibly demonstrate total regeneration of hearts in post-MI experimental rat models. Not only this, we demonstrated brain regeneration, pancreas, liver, and kidney only in Vata and Kapha mediated we were able to reverse. In Pitta mediation, we were not able to reverse. And also, what I should submit to you is that we were able to regenerate kidneys only in partial nephrectomy models. In liver, we did total hepatectomy models. We demonstrated liver regeneration, which is already, we don't have to demonstrate, it is already a very highly regenerative organ. But pancreas, no one did it. We demonstrated subtotal pancreatectomy models. We regenerated the entire pancreas. These were the results of our translation research. Next slide, please. We'll just quickly go to, I'll operate the slides, can I?
Yeah.
I'll quickly be able to finish this.
Exactly.
So, 22 years, 28,000 patients, 5 million generation of revenue. 63% of my patients who followed exactly the protocol that I mentioned have been completely free of medication. These were essentially end-stage patients who were given only 2 to 3 months of life, maximum of 7 to 12 months of life, end-stage cancers, allergies, asthma, you name it, by God's grace, we cured it. So the salient features, I keep saying this, don't violate scientific guidelines, never violate, because it's we who want to get Ayurveda integrated into modern medicine. So, if God comes onto the earth now, he would speak in English because it'd be more popular. You have to go by. Lord Krishna spoke in Sanskritam because everyone knew Sanskrit. If Lord Krishna came now, he would have spoken like Swami Vivekananda. He's not an ordinary person. He's Avatar Shiva. He spoke in English. That's how he could get the message of Bharat. So that's why I say, don't violate guidelines of that. That's because I'm a MCI licensed practitioner. I cannot go against. But even then, never violate the scientific guidelines. Target only end-stage diseases. Collaborate with the GPs. That's exactly what was being discussed today. Absolutely agree. And most of GPs increased my vocabulary. They gave me a set of so many bad words that my vocabulary increased in several languages. But several of these GPs slowly got converted, and they really send their end-stage patients now to me. And patient orientation is very important. We always tell the patient if he cannot follow all of these 5 prongs, the supplements, the diets, the fitness regimen, home remedies, yogasanas and pranayamas, specific pranayamas. It takes hardly 10 minutes for these patients to follow these yogasanas that we prescribe. Pranayamas only 10 minutes, nothing more, nothing less. And what is the result that we found? See, IDDM patients, look at the anti-GAD antibodies, anti-insulin antibodies, before and after. 4 years, it's a Vata Vikriti patient, with IDDM, and 60 units of insulin was given. 12 kilo individual kid. After 2 years, the anti-insulin antibodies come down to below 200. Anti-GAD antibodies come down to from 48 to below 10. Completely normal. His mother used to be worried about one thing. Sir, we got so used to give him insulin, prick him with insulin. Now, what do we do now? He's normal now. I said, just prick him, don't give him insulin because you're used to giving injection. So this is a Vata-mediated ischemic heart disease patient. So I gave kashayas as explained, and I gave high saturated fatty acids, food with high saturated fatty acids, and also high glycemic index foods. Calorific value, of course, I go with those. As I said, I don't violate any of the guidelines. I choose from those high glycemic index foods. So without, this is another patient. She underwent a CABG 18 months before she came to me, ended up with a AWMI. Her heart failed, 54-year-old female. Her heart ejection fraction came down to 27 after this heart attack. LED completely blocked. After 3 months, she didn't believe she was a Russian. At the age of 72, now she's still alive. She was given only 6 months to live because her EF was 27 and she was given only 6 months to live. Yeah. Completely gone. This is a Pitta-mediated individual.
EF increase.
EF increase. From 27, it's come back to 65. She was a normal individual. Kapha-mediated individual, friends. Psoriasis before, with kidney failure after 9 months. We stopped even his dialysis after 18 months. This is several cancers, more than 400 plus patients on cancer, high-grade B-cell lymphoma, You mentioned HER2 positive, PR positive, ER positive cancers. We have a lot of data. We're publishing all of them, but I have a problem with the publication. We'll talk about it later. But complete cured.
They're going to start their services.
Yeah, okay.
We're supposed to finish
Sure.
by 5. So how about we make a deal? We start where we finish tomorrow.
Okay, sure, definitely.
Yeah,
Yeah,
so that
sure.
was Dr. Polisetty on, I will start. Just before we pack up, can you just 1 minute speak about the machine?
Yeah, so what we did was we see the key here is quantification of Vata Pitta Kapha. So how do we quantify that? Through HRV, I use the HRV algorithm, created models of Vata Pitta Kapha and their subtypes using foods, and then recorded the HRV several times, and then trained the model. Okay, by the way, I'm a data scientist from University of Texas, so I learned Python programming as well, so that I can write the application for this machine. So I wrote everything from the scratch because, I could not transfer the entire domain knowledge to software engineers. I said, okay, let me learn Python. And by the way, by God's grace, it took only 3 days, friends, for learning Python. Because once you're familiar with maths and your domain expertise, Python is just, talking to the machine. So it's very, very algorithmic, friends. The key here is, just like Dr. Parashar explained, the key here is to your quantifying the VPK, the triangle. Even I made a triangle. It's interesting that, we're treading the parallel paths actually. Once this VPK is quantified in different organs, through HRV, we were able to, correlate, we induced we induced not only Vata Pitta Kapha, but their subtypes in different, in rats, because again, there are a lot of correlational sciences between the rat physiology and the human physiology. From there, we were able to correlate HRV domains. Okay, another important problem that HRV enthusiasts are actually, no one modeled Vata Pitta Kapha, and hence they're thinking that VLF is for Vata, LF is for Pitta, and HF is for Kapha. That's not right, friends. What we found was the entire Vata spectrum is spread across the entire spectrum. Pitta is also spread across and Kapha is also spread across. So we fine-tuned this HRV domains for frequency domains for individual Vata Pitta Kaphas. And then, for example, to understand the health of heart, Pranavata, Sadhaka Pitta, Avalambaka Kaphas, you have to find those frequency domains and then check their proportions. If they're in equal proportion, the heart is healthy. Otherwise,
That was Dr. Polisetty.
Oh, sorry.